Hi everyone, thanks so much for joining us today. We're going to get started at approximately 12 p.m. Eastern Time or 9 a.m. Pacific Time, so in just a few minutes. All right, we're going to go ahead and get started. So hello everyone. Thank you all so much for joining us today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I was the former NOAA Omics coordinator. Our new Omics coordinator is Nicole Miller, and she's going to be transitioning over into being your new host in the future. And I sit in NOAA's Office of Ocean Exploration and Research, or NOAA Ocean Exploration for short. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established the seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA and with our partners. The seminars from this series are made available on YouTube and posted up on our Omics website. There should be a link to our website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read the questions for our presenter to answer. All right, so with that, our presentation today is titled Making eDNA Count Towards Quantitative Metabarcoding, presented by Dr. Zachary Gold. So Zach, while I'm gonna give you screen sharing permissions. So you can go ahead and get set up. And while Zach's getting his presentation set up, I'm going to read a little bio about him. So Zachary Gold is the new Omics Group Lead at the NOAA Pacific Marine Environmental Laboratory in Seattle, leading the development and application of genomic methods to understanding how marine ecosystems are responding to warming, ocean acidification, and hypoxia. Previously, he served as a jointly appointed scientist with the Southern California Coastal Watershed Research Project and California Cooperative Fisheries Investigations, or CalCOFI for short. He led the application of eDNA efforts to Southern California marine ecosystem monitoring. Before working in San Diego, Zach was a postdoctoral researcher at the Cooperative Institute for Climate, Ocean, and Ecosystem Studies at the University of Washington and at NOAA's Northwest Fisheries Science Center. He worked with Dr. Kim Parsons and Professor Ryan Kelly to use eDNA to better understand the trophic ecology and foraging behavior of endangered Cook Inlet Beluga whales. He received his PhD from UCLA in 2020 under Professor Paul Barber, developing and optimizing eDNA efforts to survey inside and outside of Southern California marine protected areas. And in 2015, Zach graduated with a BS in marine biology with honors from Stanford University. As an avid underwater photographer, former ocean lifeguard, and National Geographic young explorer growing up in Santa Monica, he has a deep passion for coastal communities and the marine environment. So Zach, thank you so much for joining us today and presenting on your work. I'll now pass it off to you to begin.
Awesome. Thank you, Catherine. Really appreciate it. And what an honor to be here today. I'm giving the presentation to the NOAA Omics group. And I'm going to be talking about making eDNA count and the efforts in the last year towards making quantitative metabarcoding. And I just want to start by acknowledging the many collaborators and co-authors on this project. Certainly would not have been able to achieve it without all of these people's help, particularly on the Bayesian statistics and joint modeling efforts. And it's been a really fun ride. And we spent the better part of the pandemic on a computer trying to understand how to make environmental DNA quantitative. And so, the motivation, the goal of all of this work is to make environmental DNA ecosystem assessments as useful as possible, right? We want to be able to use eDNA in our marine ecosystems to give us, the kind of biomonitoring tools that we need to deploy at scale. And in order to have that kind of powerful tool, we know that abundance matters, because we can look at these two kelp forest ecosystems, and from a presence-absence standpoint, they're identical, but they have very different ecological characteristics. And the same is true of these sort of Southern California fisheries target species. We might manage these two fisheries very differently, even though from a presence-absence standpoint, they're identical, and they're telling us very different things about what's happening in our ecosystems. And so here's the problem. The problem is, if you generate a bunch of metabarcoding data and you look at it, it's kind of a hot mess. If we compare morphological counts here from ichthyoplankton from CalCOFI jars versus metabarcoding reads, like, yes, if you squint, maybe drink a couple beers, there is a correlation there, but there's a lot of noise. And in particular, the kind of noise that is striking in this image is the non-detections. And what do we mean by non-detection? It's when one method is missing it versus another. And in particular, in this study, we're going to be focused in this presentation, going to be focusing on the kind of non-detections where we have 3 technical PCR replicates, and for some reason 1-2 of those technical replicates are not detecting a given species, but the other ones are. And in those instances, it's driving a lot of noise. And so clearly metabarcoding data is very noisy. We also know that metabarcoding data is totally biased. And so if we make mock communities, we grab DNA, tissue from different organisms, we make our favorite, sort of fish community assemblage. We know the input DNA concentration, and then we go and sequence it. We don't get the exact proportions that we put in the jar. And so we know that metabarcoding data is noisy and biased. But the question really fundamentally is why? Why is our metabarcoding data noisy and biased? And we are racking our heads and trying to be able to figure out, how do we actually describe the patterns that we're seeing? And so taking that sort of step back and trying to understand why we're seeing the patterns we're seeing, start thinking about the metabarcode sausage making, right? We have some amount of biomass of organisms in the environment, and then there's the amount of DNA in the environment. And there's a bunch of different stochastic and deterministic processes that are going to impact that, from 3D transport to shedding rates to decay rates to time to species movements, morphology and physiology. And then when we have our DNA in the environment, we have to actually collect it And so there's going to be biological, sampling and different replicates might impact, sampling error, and then different collection methods. They're going to change our, efficiency of how much DNA we're collecting. And then we actually get to how much DNA, is actually, extracted. And that is just to bring it into sort of the lab. And even then, each of those individual components can be broken down into all of these subcategories, decay rates, shedding rates, transport, and assay efficacy. And each of these even subcategories could probably be broken down into another dozen or more. But then we get to the lab and we have our DNA after extraction, and we have our inhibition. We have initial subsampling that's happening because not all of our DNA extraction is actually going into the PCR. We're running our PCR, but we know PCR is biased, and different PCR methods are going to have different impacts on what we're seeing. Then we actually get our DNA after PCR. We have to do library prep. Then we have to, throw it on a sequencer. We know the sequencer is only sequencing, some fraction of the total amount of DNA that we're actually loading onto it. Then we get our DNA after sequencing, and then we have to do all the fun bioinformatics and post-processing. And then we get our data analysis. And somewhere along this very complicated process, we are, going from the organisms in the environment and to an ASV table, on our computer. And there's a lot of steps in there that can impact abundances, and it's a little daunting. But we, really focused our metabarcoding framework on, okay, what is happening in the lab and what's happening when we have DNA after extraction through sequencing, and can we understand, this source of the, the mechanism of and how, metabarcoding is working. And so here we really, focused on two elements that are controlling what we're observing. The first is subsampling. So this is, the sampling 0 is sort of you whiff. This is not surprising. This is the, marbles out of a hat kind of scenario. And really rare things are more likely going to be missed. You can have a biological 0, which is, actually a 0. The species wasn't there. There was no DNA in that extraction. And so we didn't get it, and that was correct. Or you can have, absolutely the wrong net. And so you can have a technical 0, and for whatever reason, you're not actually moving and you're not subsampling correctly, from DNA. And we also can have PCR bias. And so the combination of, subsampling and PCR bias is really what we're driving here. And in particular, the PCR bias is thinking about the amplification efficiency. And so we know that, as we're running these cycles, some pieces of DNA from some species, if they amplify better than others, can have this runaway reaction where, even things that start at a much lower concentration can end up with, much higher proportion, because anything to the power of 30 is going to have a huge impact on the number of reads that we're seeing. And so this really, this framework is what set up how our understanding of amplicon sequencing. And, we are proposing basically that signal and noise in metabarcoding data sets are a function of subsampling and PCR bias. But actually, in order to demonstrate that and prove that to you, we need to be able to model this function, and then we have to be able to test our hypotheses. And so from a subsampling perspective, this is actually pretty easy. The math isn't that complicated. Poisson distribution of the DNA molecules and the volume, this is the marble in a hat math. And we know how to do this quite well. The PCR bias, on the other hand, where do we get PCR bias? In a perfect world, PCR should double every time, but we don't live in that perfect world. Some species don't amplify at all for a given marker set, and other species amplify somewhere in the middle, somewhere between 0 and 1. You don't get that perfect doubling. And so following this, basic PCR equation, the reads that we should be seeing are a function of both the DNA concentration as well as amplification efficiency, and obviously to the power of the number of cycles that we're running the PCR at. So the idea of our mechanistic framework of amplicon sequencing is we can account for subsampling, we can account for PCR bias, we can model this, then this will set up a series of hypotheses that we could test. And there's sort of a few important notes that I want to put on the framework. First, we're explicitly stating that metabarcoding is compositional. What do I mean by compositional? I mean it's proportional. We're only measuring proportions. And so, you can get the same proportions, you can have very, very, very different absolute abundances. And so it's really important to note that metabarcoding does not provide any information on absolute abundances, because it's going to matter who else is there and as well as because we know there's amplification bias, it's who else is there and how well you amplify compared to the other species present. And we know that, these amplification efficiency biases, the observed proportions, this is pretty well documented in particularly the microbiome literature. And we also, from our work, have found that amplification efficiency appears to be consistent for a given species in a given protocol. And we're going to make that explicit assumption that it is consistent, so that we can take multiple streams of data and combine them together. And just want to note that, we've had 3 papers come out on this. Some are in preprint that just were accepted, and one in ecology on the Shelton et al. 2022 that's now available. But that model, for much of it, did not actually include subsampling. But one of our efforts did include the subsampling, and I'm going to talk about subsampling today. And so this is really, fundamentally our framework, that these 2 sort of equations from subsampling and PCR bias are explaining, what we're seeing in our metabarcoding data. And so we wanted to go out and test this. And so it's really these equations set up, sort of 4 very simple hypotheses that we can test. 1, we expect there to be more non-detections across PCR replicates for rare molecules, which is not, particularly surprising. We then also expect that we get more non-detections for species that have low amplification efficiency, which has not been demonstrated before. And then, on the flip side, on the signal side, not the noise, we expect there to be a greater number of reads for species with higher amplification efficiencies, assuming same starting DNA concentrations for both. We also expect there to be more reads for more, when there's more molecules of a given species, and for the same amplification efficiency. And so we went about testing this in 3 different ways. 1, we did a series of simulation work, on the computer. We then created a bunch of mock communities that allow us to figure out the amplification efficiencies and test some of these theories. And then we also have empirical data, and I'm going to present empirical data from 2 different eDNA data sets. One of them is from ichthyoplankton from the ethanol preserved CalCOFI jars. And this one we have both morphological identifications as well as the metabarcoding DNA. And then we also have fish eDNA from Cook Inlet in Alaska, particularly from the mouth of the Kenai and 20 Mile River. And so from a simulation perspective, this isn't really surprising, but we see that there's a much higher rate of non-detections occurring when we have really low DNA concentrations, right? We have very few of these marbles in a jar. If you're of a certain color, you're less likely to pick them up. This isn't really surprising. And so when we control for amplification efficiency, rarer molecules you have a higher chance of missing them. This is totally expected. What is incredibly and not immediately obvious is how much of a role amplification efficiency plays in creating noise. In instances where you have an amplification efficiency of about 0.5, right, that's the middle of 0 and 1 in the distribution of possible amplification efficiencies, you can still get a pretty high rate of non-detection when you're at 10,000 copies per microliter. And so this is, a huge source of noise that I don't think we as a community were really recognizing and appreciating. That this is one of the reasons why our data sets have such an incredible number of zeros in them, is because we have this PCR bias. And so we turn to our mock community data sets, so leave the computer and let's actually test this in the lab, let's test this in the field. And so when I'm talking about a mock community, we took DNA from tissues from the Scripps Institute Pelagic Vertebrate Collection, also from Northwest History Science Center, and we made a bunch of these different fish communities, and we made 9 unique communities in which were subsets of 3 of the same species, but then we changed the proportional abundance. And importantly, each of these have a unique input DNA concentration per community. And so that actually allows us to look at the same kind of figures that we just showed you for the simulations. And again, we did 3 technical PCR replicates, and we'd use the MyFish universal primer set. And the first thing to really just appreciate is the wide range of amplification efficiencies for fishes, for the same primer set. And in particular, want to highlight that it actually seem like it's explained by, the phylogenetics and the similarity in sequences, right? We're seeing species, a bunch of species in the same genus that have a pretty wide-ranging array of amplification efficiencies. And two that I want to highlight, because it's going to come back later, is that sockeye clearly has a higher amplification efficiency than coho. And that is a significant difference. I know it's hard to tell. This is, a center log ratio, so log skewed, but those are different amplification efficiencies. And so if we go and try to create the exact same figures, where on the x-axis we have the number of DNA molecules, and the y-axis we have the proportion of non-detections, we see that on the left here for the species with high amplification efficiency, we basically never get a non-detection if we have over 20 copies of DNA in the mock community. On the flip side, when we have low amplification efficiencies for some of these species, in particular, the culprits here are sharks. Sharks don't amplify particularly well for the myfish primer set, for the myfish universal teleos primer set. We see that you can have 10,000 copies of DNA and still get non-detections. And so, this is in the lab. This is controlled. We know that DNA is there, and it's not amplifying. And it's basically demonstrating that the framework and the model that we're proposing, is lining up. And again, if we look at, how many reads we're seeing for these same species for the same number of DNA molecules, we're seeing, a totally expected trend based on the framework and model. And that for species that have a higher amplification efficiency for the same number of molecules, in that tube, we are getting more reads. And we're getting, dramatically more reads, orders of magnitude more reads. And so this is lining up really, really well with our framework. Okay, so this is in the lab though. What does it look like in the real world? Well, we're not going fully into the real world. We're doing a sort of controlled experiment here where we're taking, the CalCOFI jars. These are, a really unique and interesting data set for testing this, because we have paired samples where we have things in formalin that have already been, expertly identified, and all the ichthyoplankton, by the incredible, sorting and tech team down at CalCOFI. And then we have the ethanol jars, where we can do the metabarcoding actually just from the ethanols, or sort of ethanol DNA and all the eDNA that's shed in there. And for background and context for folks who are not familiar with the CalCOFI program, this started in 1950, and really this was in response to the sardine fisheries collapse. That was a massive economic impact to the California current, and in California in particular. And so in response to try to understand, how our fisheries are changing over time, the CalCOFI program started these quarterly cruises on this grid pattern. And it's, it is in the US the longest ecological time series in the ocean that pairs hydrographic and biological data. And it's led by NOAA Southwest Fisheries Science Center, SIO, and the California Department of Fish and Wildlife. That was my previous employer until 3 months ago. And so the study design for this was we picked 4 stations that are representative of the most distinct, ecosystems and different currents and water masses that we see in Southern California Bight. We took the ethanol jars from these 4 stations, went back in time 23 years. So, when I was 3 years old in 1996, and then we did 3 technical PCR replicates each. And we used the myfish primer set, same exact protocols we did for the mock communities. And so this is the figure I showed at the beginning of the talk, where we see that if you just do the counts versus the sequence reads completely uncorrected, where there's a ton of noise and there's sort of a correlation, but it's pretty weak at best. But if we then plot the number of non-detections versus the number of larval counts we see, we're getting the exact same results as we're seeing from our simulations and we're seeing from our mock communities. And here, right, we're seeing much higher rates of non-detections for rare molecules. And here we're assuming that larval counts are directly proportional to the number of molecules of DNA, which I think is a pretty fair, assumption in this case. And clearly it's lining up, it has to be somewhat of a fair assumption because it's lining up exactly with what we expected from simulations and from mock communities. Unfortunately, we didn't have any species with low amplification efficiency in the CalCOFI data set. So unfortunate, fortunate from a data analysis perspective, because we didn't have this really occurring, but the species with low amplification efficiency were never seen in more than a handful of counts. So we weren't able to look at the proportion of non-detections for really, really abundant species. And again, if we make the same sort of similar plot with this data, we see that on average, for the same number of larval counts in a jar, species with higher amplification efficiencies, have more reads. And again, there's not a lot of species with low amplification efficiency, so we didn't have, you know, the same power as we did with the mock communities to make these kind of comparisons. But between all of these data sets, you know, I think we've sort of demonstrated pretty well that we satisfy each of these 4 hypotheses, that we get more non-detections for rare molecules, we get more non-detections for species with low amplification efficiencies, we're getting more reads for species with higher amplification efficiencies given everything else is even, and we get more reads when we have more molecules of a given species. And this is really demonstrating that our mechanistic framework is accurately capturing the patterns that we're seeing. And so the real question then is, can we actually correct for this bias and noise? And can we actually use this information to help distinguish signal and noise so we can make these sort of quantitative estimates? And so the first thing to recognize, though, is that we've been trying for a really long time to, you know, take metabarcoding reads and be like, hey, this is quantitative. But the problem is that it's a 3-parameter equation and we only have 1 data set. And so, you know, we can't solve that because there's an infinite combination of amplification efficiencies and DNA concentrations that result in the same number of reads. And so we've been, you know, barking up the wrong tree. And so we need additional information to make metabarcoding data count. And in particular, there's sort of 4 solutions that we highlight are ways to solve this problem. The first is just avoid all the math entirely and to use uniquely tagged PCR molecules. And the team in Japan, led by Hoshino et al., you know, using this QSeq method, you know, has really gone and demonstrated that if you basically add unique tags so that every time you do a PCR, it's this, you know, there's a unique primer associated with it, you can disentangle and figure out exactly what your DNA concentration was at the beginning, and you can, you know, forget the whole rigmarole. Unfortunately, it's a pretty expensive process from my understanding, but who knows, maybe it'll get cheaper, you know, because it is just generating primers, and we can, you know, bring that cost down if it gets to scale, and that's, you know, exciting element. But what we're going to talk about here, and we were really focusing on, was, you know, the other ways of solving this equation, which are to actually solve the equation instead of dodge it, which is, you know, looking at independent estimates of amplification efficiency, you know, from mock communities and from variable PCR, and also can solve this equation if we have independent estimates of absolute abundances of the targets that we're looking at. So independent, how do we get independent estimates of amplification efficiency? Well, I already talked to you about mock communities, and, you know, we're known concentrations, we're mixing these things together. The nice thing is that these are routinely demonstrated. People have been making mock communities for a long time in metabarcoding. The problem is it's completely intractable for greater than 100 species. You know, you could not make these for, you know, all phytoplankton, you know, on the West Coast, right? Or all possible zooplankton species. Maybe you could pull it off for the fishes, you know, because there's not that many species, but even still, like, having access to all those tissues and being able to generate them is very, very, very time consuming. And it's, you know, often difficult to get a sample of some of those species. On the flip side, another option is this idea of a variable cycle PCR, where we can actually capture the change in amplification efficiency. So the pro is you don't need tissues, you actually don't even need to know who's necessarily in your samples ahead of time. But on the flip side, it's only been demonstrated once. We do have data, but we have not gotten a chance to look at it to see if it worked. But fundamentally, the idea of the variable cycle PCR is to calculate the slope. You know, you stop PCR at these different cycles, and then you look at the changes in proportional abundance, and then use that to calculate your amplification efficiency, which is functionally, you know, the slope that we're seeing here. And the other way we can solve this is to look at independent estimates of absolute abundance. Now you might be saying, well, isn't the whole point we're just trying to get that abundances in the first place? So, like, why do we need independent estimates? We could just forget the whole eDNA. But there's, you know, places where this can be, you know, utilized effectively, right? qPCR, we're only targeting 1 species at a time, or ddPCR, you know, pick your favorite single, you know, molecular assay method, but we can target key species. And so we, you know, can do a combination of qPCR and metabarcoding to allow us to sort of anchor, you know, abundances to absolute abundances to a handful of species, combine them with metabarcoding data. You can also think about doing qPCR for a whole metabarcoding locus. How much is the total, you know, myfish DNA, and similarly anchoring, you know, the proportional abundances we're seeing to that. And, you know, we can also use low variance traditional abundance data as demonstrated with the CalCOFI data set. So something like microscopy, colony forming units, something like fisheries acoustics, but probably not something like beach stains, where there's an incredible amount of variability between replicates. And that'll be really hard to use to anchor the metabarcoding data. And so we can actually do these corrections though, right, with this information. So what we demonstrated in Shelton et al. 2022 was that with mock communities, we can actually improve our estimates and nail down and remove the impacts of amplification efficiency, right? We can correct for this bias and, taking the information from one mock community and applying it to 2 other mock communities with the same species. We also in the CalCOFI, work, we demonstrated that we can jointly estimate abundance. So we can use information from the environmental DNA and the metabarcoding to give us reads. We can use information from the microscopy and the visual estimates to give us DNA concentration. We can then write a joint model, estimate amplification efficiencies, get the whole model to converge, and then estimate what the true abundance was, for all the species in the jar. And here, one of the big advantages is eDNA is giving us way higher species resolution. In this case, I believe it was 15, 16 additional taxa that the microscopy can't identify. And so for really abundant things like anchovy and sardine that are relatively easy to identify, we're not providing any additional information. But for a lot of these other taxa that are really difficult to identify, eDNA provides us a way to get that data while leveraging all the visual data that's already been done. And so it gives us a more complete and accurate, picture of what the true abundances of all the fishes and ichthyoplankton in that jar are. And so from the joint model, we can see that actually we can correct for this bias, right? These are the same data sets that we showed, that had a really poor correlation, but after we correct for the amplification efficiency by developing this joint model, we actually see that we can do a pretty good job at predicting the number of counts in the jar, a much better job than before. But in particular, there's still a heck of a lot of noise, and part of this is we did not account for subsampling bias. And so that's something that needs to be incorporated in our future joint modeling efforts. And this is really obvious when you look at the observed versus predicted sequence reads. There's lots and lots of predicted sequence reads, that should be higher than 0 because other, there were other instances where one of the PCR replicates detected it, but others did not. But we are doing a good job overall of capturing the distribution and be able to use this framework and use these equations to correct for bias. And what did this allow us to do is actually go back in time and reconstruct anchovy and sardine abundances, capturing the rise of anchovy that started during and after the marine heat wave, to the point where we've had some of the highest consistent anchovy abundances that we've seen, which has been an absolute ecological surprise. And in particular, we're seeing this really interesting combinations now. They have this more complete data set of these marine heat wave driven novel community assemblages, and so we're getting these particular really unique combinations of species that have not occurred in 70 years of CalCOFI data. We're getting anchovy and southern mesopelagics at the same time. And so this goes against, all of the predictive, work from the 20th century that found really strong correlations with anchovy and sardine. And clearly that relationship is broken down. And so now the real question is, okay, well, what is driving anchovy and sardine abundances? It's clearly not a simple temperature relationship. And so some of the work that we've done in the last few months, leveraging the really incredible efforts from the NCOG program and Andy Allen's lab, and a lot of the amazing work from Chase James and Rob Lampy, is to actually how do we combine information on the prey, the trophic prey fields, from the phytoplankton, from the zooplankton, from the microbiome assemblages, and associate that with the abundance of anchovy larvae. And it's really giving us a new perspective on this idea of anchovy water and what might really matter for the success from a biological perspective of the baby anchovy, because what we've seen so far from many decades of work is that we're not quite seeing it in just the temperature and just the chemistry data. So something in the biology is happening, but obviously it's very difficult to understand what's happening in the lives of a very baby tiny anchovy. But these new molecular tools are allowing us to look at that. And in particular, really excited to start using some of these tools to look at the gut contents and see what they're actually eating, not just everything that's in the environment around them. And so then I want to present lastly like a cautionary tale of this is not a silver bullet and we're not quite there yet. But this was the hope here, was to basically start correcting for bias using a purely molecular, a qPCR plus metabarcoding approach to jointly estimate abundances. And this is just a preview, a little highlight, a taste of the work that I did during my postdoc at the Northwest Fisheries Science Center, looking at the foraging ecology of belugas in Cook Inlet. This was a big collaborative project with NOAA Fisheries and with the NOAA Alaska region, and a bunch of nonprofits and community scientists. And so, community scientists, citizen scientists are going out there with binoculars at 5 locations around Cook Inlet and recording when they're seeing belugas in both spring and fall now. And so we paired eDNA sampling along with the monitoring on a handful of rivers. We really don't understand fully, what the seasonal patterns of the fish communities are. And we know beluga are using, these areas seasonally. And so trying to get a better understanding of, who might the beluga be foraging. And one of the main motivations for this, as you probably saw from the first image, the visibility is, really, really low. And the tidal exchange is insane. The second highest tidal exchange is in Turnagain Arm in the world. It's only about like a foot to 6 inches smaller than the Bay of Fundy. There's 2,000 glaciers, so chocolate milk basically is better visibility, than the water there. And so we needed a tool to be able to start, looking at what the fish communities are, and, didn't really have a better way of doing that at scale. And so just for context, location of the world where we're working, 20-mile rivers at the end of Turnagain Arm and the Kenai River, and we're on the Kenai Peninsula south of Anchorage there. And so one of the cool things right off the bat is if we look at, our model and we do, okay, well, are we getting the same instances of noise that we're seeing? We're recreating those exact same figures. We're basically only seeing non-detections and metabarcoding when our qPCR concentrations are super, super low. But before, I think I highlighted and want to reiterate, when we had been looking at non-detections, we've been using the idea of non-detections as, when at least one of the PCR replicates are detecting it. But what happens when all 3 PCR replicates don't detect it? Well, it turns out for beluga and coho, we had a few samples and a few instances in time that didn't really make any sense. In fact, for the coho, there is an instance where there's over 1,000 copies per microliter from the qPCR. This is basically almost as high as the maximum standard. And this is true in all 3 technical replicates. And we're getting no detections in metabarcoding. And so we're like, sort of what is happening? Like, why is this breaking for these two species? Well, it turns out that all of these non-detections for coho salmon occurred in Kenai River in July. And at the same time, we look at our metabarcoding data, each of these samples had greater than 50%, almost up to 75% of the total reads going to sockeye salmon. Just an incredible number of reads going to sockeye salmon. And I'll note that all of these samples have over 150,000, copies of DNA or sequencing depth. So this is, pretty high sequencing depth for metabarcoding. This is not, an artifact. And these are consistent. All 4 of these samples are occurring in the Kenai River in July from surface samples. And for anyone who spent time in July on the Kenai River, this is probably also not totally shocking or new to you, because this is, peak combat fishing season. And there is an incredible amount of sockeye salmon that is coming up that river. And, you can have images where this is how much sockeye is in the river. In fact, it can sometimes seem like there's more fish than there is water in this river. And so clearly, in a time when there's incredible biomass of fish, eDNA is breaking down. And you can have really high concentrations, actually the highest concentrations of silver salmon and coho salmon at the same time, and completely miss it from a metabarcoding perspective. But this actually makes sense with what we know with the framework, because, sockeye salmon has a higher amplification efficiency than coho. And so we expect that this swamping is possible. We just didn't know that the swamping was possible to this degree, that you can have concentrations that are equal to your standards and still have swamping occurring. And we saw similar things for the belugas. There was less consistent of a pattern, but in all of these instances, there was a species that had 50% or more, total reads dedicated to it. And so suggesting that in instances, where there is an individual species that is, swamping, and maybe we can set a threshold of swamping at 50% of the total reads dedicated to one species, you're starting to lose detections of really common species that you can detect. Their DNA is still there from a qPCR perspective. And so with all of this information together, I think it starts giving us a picture of what we're seeing here, right? Where signal and noise are clearly a function of subsampling and PCR bias in our data. We can model this function, we can test our hypotheses, and they seem to be working out. Now the real question is, okay, we sort of know how the world works, but what do we do about it? And so that's kind of where we're at now. And so this is where the rest of the talk, I don't have great answers, and would love, thoughts and interest, because, we only figured this stuff out, within the last year, and we're still looking for answers here. What we do know, though, is that there's some really important information we need to achieve quantitative amplicon sequencing from metabarcoding, right? We need independent estimates of amplification efficiency. We need independent estimates of absolute abundance or some combination thereof. And we need to do everything in our power to increase how much total DNA we're capturing. And we also need technical PCR replication, particularly, to understand noise. From the technical replicate standpoint, we need these. Yeah, it's a question about noise and distinguishing signal from noise. That's what the technical replicates allow us to do. These are routine in qPCR studies. I don't know if you could even really publish a qPCR study at this point without having technical replicates, but it's something that has not happened in the metabarcoding community, certainly not standardized across all our efforts, even within NOAA. And obviously, it increases cost and effort substantially. I mean, if you're doing 3 technical replicates, you're basically tripling lab time and sequencing costs. And having done this, yeah, it's a real pain in the neck, and it's a lot of money. And so one of the questions, here that we don't really have a great answer to, but I think is really worth exploring is, can we use some combination of technical replicates from a subset of samples to inform the full model? Or, can you run your data set, identify, places of interest where we, really need those technical replicates, after the first run, and go back and do a subset of those? We don't know the answer there, and that's a really interesting, modeling, statistically, avenue that I think is worth pursuing. It also demonstrates that all of the efforts that we've done in the lab, in the field, to try to increase how much DNA we're getting are totally worth it. Filtering more seawater, pipetting larger volumes of PCRs. This one's an easy one to do. Just set your pipette to 3 instead of 1. But obviously, you're burning through, DNA extraction at a much higher clip. Anything you can do to concentrate DNA extractions, or even, multiple filters, pooling them, concentrating, higher yield DNA extractions. Like maybe it's worth it for that extra step, of bead beating, or doing phenylchloroform to get just every last molecule of DNA. All of these seem to really matter, and particularly matter for escaping from the subsampling non-detection area into the continuous detection. And what do we do about this too also comes down to, well, okay, I think we have a pretty good idea now of what's happening in this box, but this box is not dealing with anything prior to the DNA that we're extracting. And obviously there's a lot of work that needs to be done in this area. And one just sort of highlight that I want to recommend why the work before the lab matters is we had instances where a beluga was detected 10 miles away in Turnagain Arm, and we picked it up with eDNA. Now, to be fair, this was like a double overhead that day, tidal bore, and there is a tidal bore at every high tide, pushing an incredible amount of water, basically filling all of Turnagain Arm, every 12 hours. So it's a unique circumstance, but clearly transport matters, and transport certainly is going to matter in some areas a lot more than others. And so being able to actually understand the eDNA transport dynamics is going to be very important for our abundance. And so with all of that, sort of outlining a path for molecular quantitative abundance. What we've learned from doing the qPCR and ddPCR assays is clearly we need to always have the most abundant taxa in any sample, but also that just because you're getting the most abundant taxa, you can get non-detection of species that are there that are also hyperabundant. And so ideally, you're picking a species that's in every sample. The nice thing is you can do this after the fact. It's like maybe one of the only times in science you can cherry pick, where we can run the metabarcoding data, look at what species we have in our samples. And if there's one species that's in every single sample, wonderful, let's pick it, and use that as the anchor for the kind of work we're doing. And clearly amplification bias is real, like as we can see with the sockeye and coho in Kenai River, and clearly eDNA can be broken. And so that, there's going to be places and times and areas where we're not going to be able to get quantitative abundance estimates, unless we're, really thoughtful and going to have, the same tool is not going to work in those areas, right? And this is particularly true clearly in high total biomass environments where you can, have, if one species is super hyperabundant, it'll swamp out other species, even if they are, pretty abundant, as seen in the Kenai River. And clearly in areas of dramatic water transport, with 40+ foot tides and tidal bores, eDNA is going to give you a much different answer than in areas, that have more reasonable, tides and current exchanges. And so all of this is sort of feeding into this larger idea of a unifying theory of environmental DNA metabarcoding, where we have all of these different factors that are controlling shedding rates, all these different factors that are influencing decay, all these different factors that influence three-dimensional transport. And these together are impacting how much DNA is in any parcel of water that we might be sampling, and some combination of subsampling at multiple steps during the process and what's actually happening, in the metabarcoding in the lab are impacting, how much DNA we're getting on the far end. And there's a lot of parameters and elements in here, and clearly we are making great strides in understanding and being able to get abundance data from eDNA, but I'd be lying to you if I said we've solved this. And so what do we know, though, is we know that in particularly very simple systems like, small ponds and lakes that have pretty controlled transport dynamics, the qPCR works, quite well at getting us abundances. But we know that for really abundant species, it seems like, particularly from the qPCR and from the metabarcoding perspective, getting differences in abundances is totally doable. But there's going to be a lot of places where this is hard. Rare species are going to be very hard. Transient species are going to be very hard. Species that don't shed very much DNA, very hard. Species that don't amplify for a particular marker set, we're out of luck, use a different primer set. High transport environments are going to be really hard, and areas with high total biomass environments are probably going to be really hard. And we don't really know even what any of these thresholds are. This is just ideas and things we've seen from experience from now deploying, eDNA in marine environments for quite a long time now. And clearly, local effects matter, where you're sampling, the dynamics of your local, area are going to matter a lot. The assay design matters, and being able to use the right assay in the right place at the right time is going to matter a lot. And clearly, replication, is really important, particularly from an abundance perspective. And so, all of that putting a lot of doubt on our ability to get abundances. Well, one of the advantages, though, there's many advantages, right? eDNA is our shiny new net. It's not going to be the best tool in every system. I think that's, doing sockeye counts on the Kenai River in July, we should probably never use eDNA, and that's okay because we're already measuring that by, using a bunch of other methods. We're actually have weirs and we're counting salmon, and we've been doing that for quite a long time, and I don't think we need to change that. And so there's going to be places, though, where eDNA is our shiny new net that we should be using them. And there's still really very, very good reasons to use environmental DNA, particularly to supplement all the work that we're already doing and to start scaling up the work that we're doing in ways that, were previously completely unimaginable, right? It gives us much broader biodiversity, for the same amount of effort. And none of us have to have the taxonomic expertise between, microbes to whales to actually identify many of these species using standardized bioinformatics pipelines. These things are scalable in a way that, obviously taxonomy is not, and the potential for automation and lower cost is there and being realized more and more every year. And clearly we can get faster results if needed. qPCR results can come as fast as a COVID qPCR test if we have the infrastructure to do this. And we've demonstrated that as a nation, we can do high throughput qPCR on a national scale if needed, right? And you can imagine having, the same kind of infrastructure as one COVID center, in Seattle, but for, all of Northwest History Science Center. Like this is possible. This isn't a pipe dream. We saw this happen in the last 3 years. And also one of the cool things that it does that, we can go back in time as well as forward in time and collecting water samples. We can go to our museums, we can go to our archive samples sitting on shelves in NOAA, whether that's an ecofoci, whether it's a CalCOFI, whether that's, other archive samples that we have at Smithsonian, whatnot. And we can go back in time needed to answer a bunch of these questions where we need, 40, 50 year, 70 year, time series. And so that's sort of questions and the sort of path towards getting quantitative metabarcoding.
Great, thank you so much, Zach. That was a fantastic presentation. We already have tons of questions coming in, so folks, please go ahead and type your questions into the questions box and we'll go through each one. We have about 15 minutes. I'm going to end promptly at 1 p.m. Eastern time, 10 a.m. Pacific time. So we'll start with our first two from Denise. So would elasmobranchs amplify better with a different primer? And they also asked, how much alcohol do you need from the eDNA to run excuse me, just start over. How much alcohol do you need to run the eDNA from a plankton sample?
Yeah, great questions. I think certainly there has to be a better primer set for elasmobranchs from an amplification efficiency perspective. I don't know what that is. It actually looks like the myfish, elasmobranch also doesn't have good amplification efficiency for elasmobranchs. It just does a better job at resolving species. And so I suspect that there must be a primer set somewhere, but to the best of my knowledge, it hasn't been tested and I haven't seen it. And then from the CalCOFI jars, we are actually just taking however much liquid was off the top. That we are aiming for 125 milliliters that we ended up filtering, doing a standard Stericup, filter, 0.2 PVDF filter. But oftentimes they're, especially when there's really high volume, we had about 75 mils, and they all very high DNA concentrations. So I don't know if you could go for less total DNA, but we were doing 125 mils was more or less, like, I would say ballpark 80% of the volume in the jars, and our goal was to not disturb any of the samples whatsoever, so that way if anyone has future projects and we didn't disturb the archived samples.
Thanks, Zach. All right, our next question is from Sean. What is the effect of pooling PCR replicates and sequencing the pool only? This saves money but normalizes the impact of different PCR reactions.
Yeah, that's a really great question. I think what that ends up doing is sort of hopefully averaging and smoothing over the possibility of subsampling, but it doesn't really get to the heart of the problem. And the heart of the problem is that your different PCR replicates are, because of how much DNA you're loading into them, are going to get different species and different presence. So doing the pooling batching is sort of smoothing that variation over. But it's not going to let you distinguish, instances where, hey, you're getting high reads in, one of 3 technical replicates, which might be representative of a species with really high amplification efficiency, but actually low abundance in that sample. And so by not having the distinct separate technical PCR replicates, you can't distinguish those 2, scenarios from each other. And so, that's the advantage of having the technical replicates. But, including the pooling, we'll just sort of smooth out and try to capture more of that variation, but won't let you, answer the question about, identifying the source of noise.
Thanks, Zach. We have questions flowing in, so I hope you're ready.
Awesome.
Our next one is also from Sean. With even fishes being a complicated solve for quantitative abundance, what is your hope for using this approach in whole ecosystems, like zooplankton, phytoplankton, etc.?
Oh yeah, totally. I think something like the Luray CO1 barcode that picks up, I don't know, what is it, 35 phyla of life at this point, everything from phytoplanktons and diatoms, all the way to crabs and whales. I think there's no prayer for a marker like that, because there's just too many different factors that are occurring. And so I think, honestly, to the, another element, we were kind of hoping that we could get away with one marker for everything, and I don't think that's going to work. And I think something at the class level is the right sort of balance between breadth and specificity for a primer set. And I think there's a lot of work demonstrating that something like that works quite well. And so I can imagine, all right, if we really care about zooplankton, then maybe it's 3 primer sets for zooplankton, right? And it's what elements of the zooplankton do we really care about? But for some of these, massive universal primer sets, it's going to be really, really difficult. But I think if there are abundant taxa in those groups, we can definitely use the techniques, demonstrated here to look at changes in the very abundant taxa. But I also think that this, this work shows that for those really, really, broad universal primer sets, there can be a bunch of, species that are the equivalent of the coho and sockeye issue, right? Like there are, for whatever reason, we're just not detecting any of those species. And one thing, the recent work I know that has been coming out of the Northwest Fisheries Science Center, and Amy Van Cise and Kim Parsons, and through the Murray Project have been, working on, has found that krill, has a really low amplification efficiency for the Luray CO1 primer set. And so krill are, kind of functionally invisible using that marker set for zooplankton. And so, that's obviously a big problem, and that can sort of speak to the same kind of issues we're seeing with the fish. So I think, the more species that you're targeting at once, the more compositionality matters. And so I think, we're going to start running into problems when we start thinking about entire ecosystems at once from one marker set. And so I think, unfortunately, the solution is, be more specific and be a little narrower in what we're targeting. So maybe it's, only all decapods, for example, all cephalopods, all, you name it, sort of breaking it down to that sort of level of sort of a class distinction. I think we'll run into less of these issues.
Thanks, Zach. I do want to say to our audience folks, feel free to keep putting questions here in the question box. I don't think we're going to get to all of them before our time is up. However, as you can see up on the screen, Zach's email is here if you do want to reach directly out to him, but we'll keep going until we hit our time limit. So our next one, Zach, is from Mike. Could you speak a bit more on what makes a, quote, high total biomass environment, i.e., is a spawning aggregation in a larger oceanic area, say dense walleye pollock in the Shelikof Strait, likely to be a high total biomass setting? Or is this problem likely to be limited to river systems, small bays, etc.? And that was long, so let me know if you'd like me to repeat it.
Yeah, no, I understand that question, Catherine, and thanks, Mike, for that question. Yeah, it's something I've been thinking about, and I don't have a great, answer to it. I mean, I think I was, in particular thinking about, the Kenai River in that example where, there's just so much fish biomass. I mean, like the fish proportion to total volume in the river, I mean, it could be like, 20% of the total volume is fish at some instances, which is kind of wild. And so that's kind of where I was thinking, but definitely in spawning aggregations, I could totally imagine a situation where there's just so much gametes in the water, especially if you're sampling right in the middle of the spawning aggregation, that you're gonna be swamping and running into the same issue. And so, but I imagine that if you, came back 12 hours later after like the immediate spawning, like you might not be in that same sort of, especially in an open ocean environment or, a larger, less constrained, higher total volume, there's going to be more transport and chance for mixture and advection and dispersion. So anyways, I think I don't have a great idea of like what exactly, and I don't think there's any hard and fast rules certainly on what is a high biomass environment. But I think in any of these sort of extreme cases, where we know there's going to be a totally large amount of DNA in the environment, that on an average day, an average instance, we'd never expect there to be that much DNA. That's going to, obviously throw a wrench in our ability to get quantitative abundance estimates.
All right, thanks, Zach. Moving along, we have a question from Ian. For CalCOFI samples, would ethanol changes over time affect DNA shedding, reduced over time, and thus affect your results of community composition?
Yeah, this is something we were really interested in, and so far we tried testing for that, and we didn't find any evidence. We didn't find better correlations in more recent years than past years. Certainly, 1 year, all the data was thrown out anyway because they only did 50% ethanol and they all molded and that was nasty. So we don't talk about 1997, but I think so far we haven't seen instances of that, but we are definitely concerned, 20-year-old ethanol, are we getting, fewer species, are we getting, poorer correlations in our results? And from what we saw in our data set, no issues. I will say that the CalCOFI collection is really, really well maintained. The pelagic invertebrate collections deserve a really incredible amount of kudos for keeping those samples, in a really good state and condition. And so, it might not be true for all collections, especially if people aren't, routinely cleaning and replacing, and filling ethanol, and the ethanol is replaced too, so this isn't, this wasn't all, it was filled initially, sealed, like can't remember the frequency, and I think it changes, but every few years they're re-topping off ethanol and adding more, and we didn't notice any temporal or changes like that.
All right, thanks, Zach. We're going to do just one more question. Like I said, folks, I apologize we can't get to all of your questions, but please reach directly out to Zach if you want to follow up. This last one is from Madalena. They ask, Thank you for the talk, very interesting. Have you had experience with including internal controls, e.g. non-local fish DNA, in eDNA extractions to correct for PCR/sequencing bias?
Yeah, it's something we've thought about, but I personally haven't done any of that work. And one of the problems is that by including the internal control, that actually doesn't necessarily allow you to disentangle the amplification efficiency across all the samples. And that was something that we had long discussions with Ole Shelton about, Northwest Fisheries Science Center, and he could give you a much better answer of why that doesn't solve our problems. And so, this is why we ended up going the route of mock communities and not using the internal controls, because the internal controls basically allow you to correct for within sample, errors, but they're not going to ultimately allow you to, correct for the amplification efficiency bias across all samples, because every sample, presumably, if you're sampling in the environment, like every sample is going to have a different species assemblage over time. And you won't have those every single possible permutation of intercalibration with those samples, and so it's really difficult, basically impossible to actually use those to get the kind of independent amplification efficiency estimates. Not saying they're not useful. I think they're particularly useful for identifying where sequencing errors are happening or where individual samples maybe failed and had something weird happen in PCR library prep. And so I know that previous studies have used those internal controls as basically a quality control check, and so I think they're very helpful for that. But in terms of using those for quantitative abundance corrections, I don't think that that's possible.
Awesome, thank you so much, Zach. We are going to go ahead and wrap up our webinar. So Zach, just want to thank you once again for presenting today. We really appreciate you making some time to talk a little bit about your research. And like I said, if folks have further questions, just go ahead and follow up directly with Zach. All right, so with that, I also want to thank everybody for attending our seminar today. We actually have another seminar coming up very soon. It's on March 1st at 12 p.m. Eastern Time. Our speakers at that time will be Mark Stokel and Jesse Osbel from the Rockefeller University, and their talk is titled Marine Fish eDNA Metabarcoding, Promising Developments and Early Applications. So the registration for that is up on the NOAA Omics website, which is linked in the chat. So please register. If you have questions, don't hesitate to reach out to us. So thank you all, and we'll see you at the next webinar.